So this is a mouse. Imagine if you could take the adult cells from this mouse and somehow reprogram it into a pluripotent stem cell that could eventually develop into any type of cell in the body. Well, that's exactly what this man, Shinya Yamanaka, did in his lab in 2006. In his experiment, he forcibly expressed certain transcription factors in isolated fibroblast cell populations, which induced properties of embryonic stem cells. He then took the cells and injected them into blastocysts, which then was shown to become pluripotent. These cells were in the end called induced pluripotent cells. But what were the transcription factors that were used? They were OCT4, SOX2, c-MYC, and KLF4. In this video, I'll be focusing on the protein OCT4. Also known as an octamer binding transcription factor 4, is a protein in the POU family that is encoded by genes involved in the regulation patterns of development. OCT4 are DNA binding proteins that regulate specific gene expression by binding to ATGC-AAAT motif on the enhancer and promoter regions of DNA. The expression level of OCT4 is crucial for the maintenance of pluripotency and early cell differentiation. If that wasn't clear, imagine each individual block in this Lego house as an individual adult cell. And then this happens. If the kids in the video were representative of OCT4 protein, then the Lego blocks that used to be specialized into holding the building together is now a free Lego block that can be used to make anything, similar to a pluripotent stem cell. The structure of OCT4 is definitely an interesting one. OCT4 has two DNA binding domains, which consist of POU specified domain, or POUS, and the POU homeodomain, also known as POU-HD. Looking at the overall structure, the two subdomains of OCT4, POUS and POU-HD, are connected by a linker region of variable length and composition. Together, they have protein residues that are within 5 angstroms of DNA, like K94, or some protein residues that are in direct contact with the protein residue itself, like Q44 and C48. The POUS domain and the linker region are important for maintaining pluripotency, while the POU-HD domain is specialized in bringing the OCT4 protein into the nucleus. In addition to binding to the octamer site of ATGC-AAAT, the POU domains also bind to the palindromic octamer recognition, also known as POR. POR exists on the enhancer of the OPN gene. Although OCT4 is also shown to bind to POR as a monomer, the activation of the enhancer on the OPN gene usually requires 2 OCT4 structures. That's why in the following picture, and the structure provided by the protein database, there are a total of 4 separate domains that can be seen, which exist among 2 OCT4 protein complexes. While OCT4 binds to DNA as monomers, they can also bind to other transcription factors such as SOX2, which can generate a greater variety of cellular processes. For example, in this graph, OCT4 binds to other transcription factors like SOX2, KLF4, NANOG, and other complexes that eventually produce the pluripotency gene. Looking at the structure of OCT4 extracted from the protein database, we see a DNA helix running in between the OCT4 complex. There exists 2 OCT4 complexes with the blue-green colored structure as the POUS domain and the yellow-orange colored structures as the POU-HD domain. On the OCT4 complex, there exists a large number of protein residues interacting with the DNA. Here, we zoom in on a specific protein residue, glutamine 44, which makes direct contact with the DNA helix. Here, we zoom in on another protein residue, glutamine 27. However, this time, the protein residue does not directly come in contact with the DNA, but rather resides around 5 angstroms away. Although the reason why OCT4 induces pluripotent stem cell characteristics is still unclear, we know that the OCT4 transgenes incorporate themselves into the host cell DNA before reprogramming into induced pluripotent stem cells. Since the discovery of the function of OCT4 in converting adult fibroblast cells into pluripotent stem cells in mice, scientists have attempted to recreate such a phenomenon in humans. Unlike mice cells, which used OCT4, SOX2, C mice, and KLF4 to induce embryonic stem cell characteristics, humans use distinct 4 transcription factors, OCT4, SOX2, NANOG, and LIN28, to induce embryonic stem cell characteristics. Who knows? The ability to reprogram cells may help cure disease and recover damaged cells in specific organs in the near future. Because of the discovery of this unique function of OCT4, Shinya Yamanaka won a Nobel Prize in 2012 in physiology or medicine and helped the world take a huge leap in the field of medicine.